2 weeks ago, China made history with the first use of CRISPR gene editing on a live human subject. Some think it could set off a biomedical race to harness the technology, but with any new technology, there is the possibility that it will be misused. That will be the topic of a day-long symposium today at Dalhousie's Medical School. Francoise Baylis and Paola Marignani are co-moderators.
Ms. Marignani, welcome.
Thank you.
Ms. Baylis, welcome. Thank you both for coming in so early this morning. If we could begin with you, Paolo, give us the quick layman's definition of CRISPR.
Well, CRISPR is the modern-day version of gene editing. It's basically leveraging the bacteria's ability to fight off viruses. And we've been able to capitalize on that technology and use it in human cells and animals. And now China and the U.S. are both racing to do CRISPR in humans.
What's the significance of that?
The significance of this technology is that we do have the potential to cure diseases. Not all diseases for now, but the Chinese and the Americans are hoping to go after non-small cell lung carcinoma and melanomas and some sarcomas. So we do have the potential to change the disease.
Does it seem promising to you?
As a scientist,
yes.
Yes. So I've taken this technology and I've incorporated it into my own sort of toolbox of technologies of gene editing. So for me, it is promising. Although I think scientists in general are cautious when we move forward with these types of technologies that could have significant impact not only on our species but on all species in the planet, the genome of the planet, so to speak.
And ethicists have some concern because the last time we talked to Ms. Baylis, I think you were in Ottawa or Toronto for a conference on some of the possible negative impacts of CRISPR.
Right. So I think one of the things that people want to look at from an ethics point of view is both what we would think of as the promises and the pitfalls. And so we recognize with all science, not just this, that there are ways in which we anticipate and we intend to use them for the benefit of all. And you've heard about that in terms of developing possible treatments. But people also worry about what are all the other kinds of ways that it might be used. And aside from issues of intent, I think a lot of us are also concerned about situations where you have all the right intentions, but things can go wrong.
Mm-hmm.
One of the things that it seems to me that you were looking at the conference, this is back in the spring, I think, was it's one thing, I'm a little past my reproductive best, but let's say I were a few years younger and still in
Just a few.
Yeah.
I'm just going to stay out of this
Okay,
conversation.
properly so, okay, like a couple of decades. But that in fact, if Paolo were treating me for some disease, that in fact it's all well and good if it's going to only affect me as opposed to my progeny. Isn't that's part of the issue, isn't it?
Right. So there's two ways in which that could become an issue. So right now, the intention in terms of developing treatments is to cure patients with underlying illnesses. And we've heard about one, cancer, another one might be Huntington's disease. And so basically the idea is you have an underlying health conditions, we want to help you, and we try to treat you. Well, one possibility is we would unintentionally affect your reproductive cells. So we would affect your sperm, in your case, or in the case of a woman, we would affect the eggs. And then the concern is that whatever change or manipulation we've made, such that it results in a change in you, would also be a change passed on to your children.
And beyond.
And beyond, that's correct. So it carries through the line then. Mm-hmm. And so that becomes an issue in two contexts. One, when it happens accidentally in the context of treatment, but two, when it might actually be happening purposefully in the lab because people are taking the reproductive cells and the human embryos and purposefully manipulating them with the idea of, for the sake of argument, improving the species. And now you can understand improving in lots of ways. It might just mean improve by not having the genetic illness carried on for generation after generation after generation. But the other issue, which certainly people in ethics talk about, is what happens when we can actually improve, meaning enhance, make them superhumans.
Yeah. And I'd ordinarily ask you about whether or not scientists are concerned as opposed to ethicists, but I could tell by your reaction what she had to say. It's a concern in the lab as well as in the ethicist's office.
Clearly my poker face is not very good.
Not working this morning.
It is true. I think there is fear for the potential of creating superhumans. However, at this moment in time, scientists don't even know which genes are responsible for all the genes responsible for muscle strength, for example. And so it'll be a fair amount of time before we arrive at creating these superhumans. We've just started the first clinical trials starting in China at this moment in time. So I think there's
what did they actually experiment when the Chinese experiment of last week? What were they doing?
They have 10 patients that have been recruited with non-small cell lung carcinoma, late stage, and what they do is they remove the T cells. So this is a type of blood cell from the patient. They CRISPR or re-engineer a specific gene in that blood cell that protects the cancer cell from being attacked by the patient. They remove this gene by CRISPR, then they grow up these cells, and then they return the cells into the patient. And in essence, they've made the patient able to attack the cancer better. And so we don't know what stage it's at right now. We just know that they remove the T cells and introduce the CRISPR-Cas technology. So this is very early stages, and so although I appreciate some of the fears and the superhumans, super soldiers, and the weaponization of CRISPR-Cas, but I think there's a fair amount of time before this could happen. And I do believe that scientists are concerned about the technology as well. I mean, this goes back to Nobel. The reason why the Nobel Prize exists is because when he invented dynamite, he realized the danger of dynamite. Although he invented it for good reasons, he himself identified the problems. And I think the scientists who invented the CRISPR-Cas technology that we're using today also we're cognizant of the fact that this could potentially be used for bad things.
Yeah, and if I can just weigh in there, I think that's absolutely right. And, one of the American scientists, Jennifer Doudna, who, is key to the development of this technology, has been front and center in terms of trying to name these issues and trying to get her colleagues together to have debate and conversation. And in fact, I will be going to a meeting, a small invitational meeting with her in January and a team of scientists, because we really are trying to think in advance, how can you direct the science for the benefit of all?
Before the fact rather than after the fact.
Yes.
Exactly.
Well, I'll have to leave it there for this morning. Thank you both very much for coming in early this morning. It's a pleasure to see you both.
Much.
Great, thanks.
Françoise Baylis is Canada Research Chair in Bioethics and Philosophy at Dalhousie. Paola Marignani is a cancer researcher at Dalhousie School of Medicine. The Royal Society of Canada Symposium on Gene Editing runs all day today at the Sir Charles Tupper Building, and all are welcome. We'll tweet out a link to the schedule.